Our protocol allows researchers to study the vessel network formation process in a clear and easily trackable fashion, opening the doors to new studies and shedding light on blood vessel behavior.
This technique presents a possibility of creating highly organized and repeatable vascular networks which respond to the surrounding environment.
Endothelial cells from patients suffering from a vascular disease can be retrieved and used in this system, making it possible to recreate the vascular disease and find an appropriate treatment.
The proposed method can be extended for studying vascular network behavior when paired with different cell types, allowing to observe the interaction of developing vessels with a specific tissue type.
Begin by submerging the scaffolds in 70% ethanol for a minimum of 15 minutes, then wash them twice in PBS. Mix 1.5 microliters of fibronectin stock solution with 28.5 microliters of PBS per scaffold to be seeded. Prepare 1 fibronectin dilution to be used for all scaffolds to avoid pipetting errors. Place the scaffolds sparsely on top of a hydrophobic surface and cover each scaffold with 30 microliters of the fibronectin dilution. Replace the plate's lid and put the fibronectin-covered scaffolds into an incubator set to 37 degrees Celsius and 100% humidity for a minimum of 1 hour. After incubation, lightly rinse the scaffolds in PBS to remove fibronectin remnants. The scaffolds can be kept in PBS at 4 degrees Celsius for up to a week. Prepare endothelial cell or EC medium by mixing the basal medium with the corresponding medium kit components, including an antibiotic solution,